Mr. M. And here's your host, Mr. M.
Hey boys and girls, have you noticed that the trees are starting to change colors? Have you ever wondered why they change colors in the fall? Well, with the lab techniques that we learned this past week, we're going to find out. Last week, we were introduced to a technique called paper chromatography. Chromatography is a way to separate components of a mixture. The first thing we did was prepare our chromatography paper. We drew a line at the bottom to mark where our samples started. We marked where our samples would be placed. Next, we use capillary tubes to put our samples onto our paper. We used a couple samples at a time. We then allowed our samples to dry on the chromatography paper. We placed our chromatography paper in a beaker with solvent. A solvent is a liquid that will dissolve our samples. We also placed a lid over the beaker. Over time, we saw that the solvent traveled up the paper. Our samples also traveled with the solvent. We also noticed that different pigments traveled at different speeds. When the solvent was close to the top of the paper, we took our chromatography paper out of the solvent. We then marked where the solvent traveled to and allowed our samples to dry again. We measured how far each component traveled with the solvent. We used metric units, of course. Knowing how far known samples traveled allowed us to identify them in unknown samples. In our first experiment, we looked at some common food dyes. We found that each food dye traveled a different distance on our chromatography paper. We also found that some dyes, like cheddar cheese, were made of a combination of food dyes. The food dye cheddar cheese was actually made from yellow 6 and yellow 7. We were commissioned by the FDA to determine whether a new cough syrup was using an illegal food dye. We did this by running the cough syrup dye on our chromatography paper. Using chromatography, we were able to determine that the food dye in the cough syrup was made of a blue dye, which was legal, and a pink dye, R3, which is illegal. Next, I commissioned you to be a part of an investigation. Someone had stolen my cat and left a ransom note. I needed you to use chromatography to be good detectives. Additionally, someone left a note to the principal saying that I had resigned. This also needed to be looked into. Using handwriting analysis and chromatography of ink pens, we were able to catch the crooks. Now that we've reviewed what we learned this week, let's return to next week's driving question. Why do the leaves change colors? In your notebooks, jot down what you think the answer is. Remember that you can always revise your ideas as you learn more. In order to answer the driving question and understand next week's lab, you'll need to have an idea of what photosynthesis is. The word photosynthesis comes from two root words, photo, meaning from light, like a photograph, and synthesis, which means to make or synthesize. Together, photosynthesis literally means to make from light. You should have learned that plants need carbon dioxide from the air and water from their roots to survive. Plants are able to use carbon dioxide and water and the energy of sunlight in order to make starch and oxygen. Starch is a type of sugar that plants use to grow and to live. In future labs, we will explore photosynthesis in more detail. But you may have heard that chlorophyll, a pigment in plants, plays a major role in photosynthesis. Now this bush here has been red all year. Does it have chlorophyll in it? I thought chlorophyll was green. What does it use for photosynthesis? In this week's lab, we will use chromatography to explore plant pigments and see if chlorophyll really is the only pigment plants need to survive. Homework, I'd like you to pick 4 or 5 leaf samples from around your area. Try to get some red samples, some yellow samples, and some orange samples, but definitely get some green leaves as well. We'll be using our photo paper to make impressions of these leaves, and then we can put those in our notebook. But we're going to use the leaves for the lab on Monday. In the lab Monday, we'll be using your leaf samples. You'll have a couple different kinds and a couple different colors. We will be using a mortar and pestle. This is a tool that we can use to grind up our leaves. Next, we will be using the jars that we used last week for chromatography. We will also be using chromatography paper. For our solvent, we'll be using rubbing alcohol again. Remember that rubbing alcohol is flammable, so we'll have to be careful about using it. Like last week, we'll also be using capillary tubes to put our sample onto our chromatography paper. We will start by ripping up our leaves into small pieces. Next, we will add a little bit of solvent to our leaves. The solvent will allow us to crush our leaves into a liquid. It will also dissolve the pigments within the leaves. You'll want to grind your leaves for several minutes. The resulting product should be a dark green liquid. Now that we have the plant pigments dissolved in our solvent, we can use this mixture for our chromatography paper. Using a capillary tube, transfer some of your liquid to your chromatography paper. Make sure that you transfer enough of your mixture to the chromatography paper. You want your sample to be dark and concentrated. Make sure to let your samples dry. Repeat the chromatography procedures that we did this past week, putting a little solvent in the bottom of your beaker, and then putting your chromatography paper in the chamber. Now that we've gone over the procedure for Monday's lab, we're ready to do the investigation. Please remember to write in your journals and bring your leaves to class on Monday.